Hello. I hope everybody's having an absolutely wonderful day. This is chapter 17 and it is on blood. And this is the first of probably 5 parts. And this is going to focus on an introduction to blood. So, first of all, why does it matter? Well, understanding the anatomy and physiology of blood is going to help you to advise patients on activities to prevent blood clots during things like hospital stays. Now, blood is a life-sustaining transport vehicle of the cardiovascular system. Blood is moving, right? It's always moving. Then we have the functions of blood, and they include, first would be transport, and then we have regulation, and then we have protection. And I'm going to talk about each one of these individually. So transport. So blood's going to transport oxygen to the cells of the body, nutrients to the cells of the body. It's going to transport metabolic waste like carbon dioxide to the lungs, nitrogenous waste to the kidneys for elimination. It's going to play a big role in the last chapter we just did, chapter 16, because it's going to transport hormones from endocrine organs to target organs. Then we have regulation. Regulation functions include to maintain body temperature by absorbing and distributing heat. So if you think about it, when we get cold, we have vasoconstriction occurring to kind of keep our body temperature core, stable. But if we're really hot, then we vasodilate, and then we try to kind of dump heat to the environment, if you would. We have maintaining normal pH using buffers, and then it maintains an alkaline reserve of bicarbonate ions, and then maintaining adequate fluid volume in the circulatory system. And then finally, we have protection. And protection functions are going to include things like preventing blood loss. So the second, I think probably the fourth segment in chapter 17 is going to be on clotting, and basically kind of like wound healing, but not epithelial, more like in the vessel healing. And we have plasma proteins and platelets in blood are going to initiate the clot formation. It's actually pretty complex, but we'll go over it. And then preventing infection. And this is because agents of immunity are actually carried in the blood. And these agents of immunity are antibodies, complement. And then the one that we'll actually talk about in this chapter, which are the white blood cells. We'll talk about antibodies and complement when we get to the immune system. Blood is the only fluid tissue in the body, and it's a type of connective tissue. The matrix is non-living fluid called plasma, and then cells are living blood cells called formed elements. The cells are suspended in plasma, and the formed elements include erythrocytes, or red blood cells, or RBCs, leukocytes, or white blood cells, or WBCs, and then platelets. Now, if you were to, let's say, take blood from a patient and then spin it in a centrifuge, the spun tube of blood is going to give you 3 layers. And when you centrifuge blood, excuse me, what you're doing, or when you centrifuge anything, is you're separating it out based on weight. So what ends up on the bottom of the tube are going to be the erythrocytes, and it's going to be about 45% of whole blood. And then hematocrit is a term that we use to mean percent of blood volume that is red blood cells. And then the normal hematocrit for males is going to be somewhere around 47% plus or minus 5, and for females about 42% plus or minus 5. Then sitting on top of the erythrocytes, we find the buffy coat. And this is going to be less than 1% of whole blood. And this is going to be composed of white blood cells and platelets. It's going to be a thin whitish layer. Again, it's going to be above the red blood cells, and it's going to be below the plasma. The plasma is going to sit on top. It's going to be about 55%. And then I think on the next slide we talk about plasma, but let's see. So take blood, put it in a tube, spin it, and then we end up with the erythrocytes on the bottom, about 45%, the little buffy coat, which they're making way bigger than what it would be probably in real life, and then the plasma would sit on top. Oh, we talk about blood first, then plasma, yes. So blood is a sticky, opaque fluid with a metallic taste, and it's going to have a different color depending upon the oxygen content. If there's a high oxygen content, it's going to be a scarlet red. If there's a low oxygen content, or low oxygen levels, I should say, then it would be dark red. The pH is going to be somewhere between 7.35 excuse me, and 7.45. And our body does maintain that blood pH in those relatively narrow parameters. And it makes up about 8% of body weight. And in males, we have about 5 to 6 liters of blood, and females about 4 to 5 liters of blood. And then plasma. So plasma is a straw-colored sticky fluid. It's about 90% water. And then there's over 100 dissolved solutes in plasma. And this is going to include things like nutrients, gases, hormones, waste, proteins, inorganic ions. But the most abundant are going to be the plasma proteins. And the plasma proteins are going to stay in the blood. They're not actually taken up by cells, and they're produced mostly by the liver. The plasma protein that is the most abundant is going to be albumin, and this is going to function as a carrier of other molecules, as a blood buffer, and it's also going to contribute to plasma osmotic pressure. This is just a table that I left in here in case you wanted to look at it. It's showing different plasma proteins, basically composition of plasma in this particular table. So you can see we have electrolytes, the different proteins, and then nutrients, gases, hormones. And then we start talking about the formed elements. So the formed elements are red blood cells, white blood cells, and platelets. Now, only the white blood cells are complete cells. Red blood cells do not have a nuclei or other organelles. And platelets are just cell fragments. Most formed elements survive in the bloodstream only a few days, and most originate in the bone marrow and do not divide. So this right here would be an erythrocyte, or a red blood cell. This would be a leukocyte, or a white blood cell. And then this would be more of what we would see in the lab. And so the little pink ones here are going to be your erythrocytes, or your red blood cells. And then these are going to be your leukocytes. And it's actually showing you different ones: neutrophil, lymphocyte, eosinophil. Don't worry about that in this talk. It'll be in the next, not this talk, not the next one, but the one after that. And here is, oh, it says questions, but it's actually a question. All right, that's all I have for you for this talk.